today's topic is structure and functions of immunoglobulin. Now, you have been studying for the past several lectures that B cells are the only cells that make immunoglobulins as cell surface receptors, and the same cells start secreting antibodies once they are differentiated to plasma cells. Now, it was already known that there is a soluble component in the blood which can counteract very effectively with the immunogen or pathogen as it was known earlier, and this was known already beginning of the 20th century. People since then were trying to look at or identify what is this molecule or what are these molecules that participate in the humoral immune response. It was around 1939 that Tiselius and Cabot were the first to report that this component antibody is present in the gamma globulin fraction of the serum. Now, technology was primitive those days, and the only method by which scientists could identify which were the fractions present in the serum was electrophoresis, simple electrophoresis, where proteins could be separated based on their net charge. Now, Tiselius and Cabot did a very simple experiment, and it took them several months, perhaps also years to do, is they have made antiserum to ovalbumin. When they electrophorese the antiserum, as is known for even just serum from any individual, there are essentially two sets of proteins that one can identify: albumin and globulins. Now, globulins also could be separated, as can be seen on this graph, into alpha, beta and gamma. Now, when they have taken the same aliquot of antiserum from rabbit raised against ovalbumin, that is egg albumin, to which now they added a certain amount of the antigen ovalbumin. Now, they already knew that on addition of antigen to antiserum always or most likely resulted in precipitation, precipitates that could be centrifuged down to a pellet. So, they did that. Now, they have taken two aliquots, like I said, one where they have not added the antigen and the other one where they have added the antigen. After what they thought was appropriate antigen antibody interaction, they centrifuged down the particulate material, which was nothing but the immune precipitate, and now they have run electrophorese both the samples on their native gels, where proteins were separated as albumin and globulins alpha, beta and gamma. Now, what could be seen very clearly that in the aliquot which showed precipitate on adding the antigen, there was a decrease only in the gamma globulin fraction, and it was quite clear. You can see the decrease. Therefore, it was easy for them to then conclude that the immune response that one can generate by way of secreting antibodies in the blood resided in the gamma globulin fraction, and therefore, the name immunoglobulin came about. So, it was Tiselius and Cabot who showed for the first time that antibodies were gamma globulin fraction. Since then, people started to do more search on how to find out the structure of this gamma globulin fraction or the antibody that we know now. Now, there were large number of scientists who were looking at the structure, and now we know the structure as two heavy chains which are held together by disulfide bond, which in turn hold the light chain. But it took very many years for Porter and Edelman to come to this, just because the technology then was not as advanced as it is today, and they always needed to work with milligram amounts of the molecule. Anyway, this picture here shows the results of two of the experiments, the major experiments. One where they have taken the antibody molecule immunoglobulin and now reduced the disulfide bonds with mercaptoethanol. Always this resulted in identification of two bands, they called heavy and light. They were always two bands, but when they looked at the size of the tube, they realized immediately that the structure should be much larger. Now, the size of the heavy chain is only around 55 kilodalton and the light chain is around 25. So, therefore, and they knew that the whole protein molecule was around 1,50,000. Therefore, they could do simple calculation and surmise that the immunoglobulin molecule is made up of two heavy chains and two light chains. Now, in addition to these experiments, they also were digesting the proteins with different enzymes, and they were able to come to a good conclusion with respect to the structure by digesting the molecule with pepsin or papain. Interestingly, pepsin digestion attacks the site on the immunoglobulin molecule, which is after the disulfide bonds which hold the heavy chain together. Papain, on the other hand, cleaves the molecule at a site which is above the disulfide bonds. Therefore, the pattern of the fragments that they obtained were quite distinct. What they obtained with pepsin digestion had a fragment which could bind two molecules of antigen, whereas with the digestion with papain, they got a fragment which could bind only one. It was monovalent, only one antigen molecule. Now, upon papain digestion and pepsin digestion, they were able to get fragments which they called Fc, and we know that this is the constant domain of immunoglobulin. Now, putting all these observations together, they came to the conclusion that the antibody binds two antigens from the amino terminus, and that antibody molecule is at least bivalent. Now, this was quite remarkable considering the time at which they were working in trying to elucidate the structure of immunoglobulin. Now, already in the 1930s, sequencing of protein, the methodology at least, came to be established. So, scientists started to sequence immunoglobulins. Now, how were the immunoglobulins purified? By the method of either salting out with ammonium sulphate or by electrophoresis, as I have shown in the first slide. But this was always from the immunoglobulins from serum. Now, they could not come to any conclusion with respect to the sequence. Now, of course, it is very easy for us to see why, because there is tremendous variability at the N terminus, and therefore, the N terminus sequencing was not possible due to tremendous variability at the, this should be N terminus, variability at the N terminus. So, how did they then get the sequence of immunoglobulins? Now, you know that in the database we have large number of sequences of various immunoglobulins. So, it was after several years that scientists found, in fact, immunoglobulins could be sequenced because of the discovery of myeloma proteins. Now, what are myeloma proteins? Myeloma proteins are products of cancer of plasma cells. Now, myeloma proteins are also monoclonal. Why? Because cancer is usually monoclonal. Almost any cancer one can think about, whether it is epithelial or humor, let us say from hematopoietic lineage. Cancer is usually monoclonal. Now, if a plasma cell gets transformed, plasma cell, remember, the cell that secretes antibodies becomes immortal now or becomes cancerous, then this cell and the progeny of this cell would make antibodies of only one class, only one type, and those proteins were known as myeloma proteins. Of course, at that time when myeloma proteins were discovered, it was not known what are these molecules, they came to be known later. So, what are myeloma proteins? They are secreted immunoglobulins from the cancer of plasma cells. Now, at this junction, though it is not really relevant, I will also like to say that people were also able to identify Bence-Jones proteins, which were not the whole molecule of immunoglobulin, but light chains. Now, myeloma patients secrete in their circulation not only myeloma, that is the whole molecule immunoglobulin, but they also, some of them express large amounts of the light chains, and those were called Bence-Jones proteins, until finally anyway they realized is a whole molecule myeloma or Bence-Jones light chains alone. Now, soon after that came to be known, almost every myeloma protein that could be identified was sequenced. The amino acid sequences then were analyzed, and it was found the following observations were made, that the amino termini of the immunoglobulins, that is 1/3, were very variable. So, when they looked at all the sequence of the myeloma proteins that they had now a bank of, they found that the amino termini always were very variable, and therefore, it was logical to conclude that this region would constitute the antigen binding region. 2/3 of the immunoglobulin sequence towards the C terminus comprised of common sequences. Now, when they looked at those common sequences, they could see that immunoglobulins, or well, these myeloma proteins then could be categorized into 5 main classes, and we know them as IgM, IgD, IgG, IgE and A. Now, once the sequences came to be known, on closer look of the variable domains, these are the variable, there are now 2 graphs here that I before me. One shows the variable domain of the heavy chain and the other one variable domain of the light chain. Now, when people analyze these variable sequences closely and they tried to see how variable are these variable regions. Now, they did a very simple analysis, and which is shown here. Now, the variable domain, first of all, they could see was around 110 amino acids from any and every myeloma protein, okay, both heavy as well as the light, about approximately 110. Now, they made these graphs by putting on the x axis the amino acid number, and then they tried to look at how many times, how different is that particular the amino acid at that particular residue, that number. So, y axis shows the variability, which would mean that the peaks that you can see here would mean in the position now, in this particular case where it is maximum, let us say in the 94th position or 95th position, the amino acid varied almost more than 100%, which would mean that this would be the most variable region and residue, and definitely this would contribute tremendously to the antigen binding. Now, another observation that in fact you can also make looking at this, that both in the heavy and the light chain variable region, there is a hypervariable, or there are hypervariable regions. Look at the residues from around 35 to 38, similarly just let us say 50 to about 55, and similarly around 90 to 94. Now, these varied tremendously in all the myeloma. Now, these three regions are flanked by more or less constant domain. Of course, there is a region here which is more variable than the others. So, they coined the terms that these were the CDR 1, 2 and 3. CDR stands for complementarity determining regions, because by then they already knew that these were the regions by which antibodies or B cell receptors bound their cognate antigen. Now, flanking these are the framework regions, which remain more or less constant. Now, in fact, today it is possible to design primers corresponding to the framework region and amplify the hypervariable domain, okay, because one cannot determine or design primers to get only the hypervariable domains. Now, now let us look at the heavy and the light variable domains, and you can see immediately that in both cases the CDR3 is most variable, and overall the heavy chain domain is much more variable than the light chain domain. The CDR2, for example, of the light contributes minimal to the antigen binding pocket. So, the H chain contributes more to the antigen binding pocket than the L chain. Now, people have done various experiments even at the protein level, trying to see whether when you separate the heavy and the light chains, whether they are still capable of binding the antigen. And almost with about 95% cases, when H heavy chain is separated from the light chain by simple reduction of the disulfide bond, the heavy chain still can bind the antigen, whereas light chain almost cannot. Now, when of course, I will like to mention here that the heavy chain separated from the light chain still exhibits binding to the antigen, but the affinity with which the heavy chain still binds could be 100 and sometimes maybe 1000-fold lower. Now, here after all the information that was gathered with respect to the amino acid sequence and after reduction of the heavy and light chain and the bindability and all the data now could be put together to come to the final structure of the monomeric immunoglobulin. Why I say monomeric is because there are classes of immunoglobulin that show oligomeric structure. This is a basic structure of an antibody molecule, or you can say immunoglobulin molecule. What is the structure? We know very well now that every immunoglobulin is made up of two heavy chains which are held together by disulfide bonds. Now, the heavy chains themselves are again held to or hold the light chain through disulfide bonds. Both heavy chains of an immunoglobulin molecule are identical, so also the light chains. An antibody molecule is at least bivalent. Now, in comparison to those which have more than one monomer, where of course the valency can go as high as 10, and we will see that in a while. The heavy chains could be either mu, gamma, alpha, delta or eta, and the antibody molecule, the class, is known by the heavy chain. So, M, G and so on. the kappa light chain could be either, sorry, the light chain can either be kappa or lambda. It's important to know that apart from the heavy chain being identical to each other, even the kappa would be identical not only with respect to the variable domain, but also with respect to the constant domain. Remember, in my last lecture I did mention that always the recombination of the kappa light chain allele 1 starts first, and if that fails, the second allele takes over. If the recombination of the second kappa allele also is disaster, then the lambda allele 1 starts the recombination process. So, therefore, we know that an antibody molecule always has only one type of heavy chain, one type of light chain. I should have mentioned something about the constant domains here, which I forgot, and let me go back to the basic structure again, that the variable domain and the the constant domains in fact are almost similar with respect to the number of amino acids. The heavy chain after the variable domain has 3 constant domains in 3 out of the 5 classes of immunoglobulin that we just talked about. The constant domain of the light chain is only 1. Immunoglobulins are glycoproteins, they are glycosylated. The glycan chain normally is present, or the molecule, the amino acids that get glycosylated are on the constant domain 2 of the immunoglobulin molecule. Both the heavy chains are glycosylated. A structure, this is a structure which shows the fold of the molecules. It is the same immunoglobulin molecule, but this elaborates the function of the intra-disulfide bonds, and let us look at this a little bit closely. You can see very clearly from here that the heavy chain has 1, 2, 3 and 4 folds. These are called immunoglobulin folds. In fact, there is a superfamily where immunoglobulin is the prototype molecule, and this family, I can give you a few examples. The alpha 1 beta 1, the co-receptor for the immunoglobulin, the B cell receptor, or the CD4 or the CD8 or the TCR receptor and the co-receptor, all of these belong to the immunoglobulin superfamily, and all of them would have perhaps different sequences, of course, at these different regions, and the structure may be very different, but all of them are characterized by the presence of these immunoglobulin folds. These immunoglobulin folds are held together or stabilized by intra-disulfide bonds. So, you can see the complexity of the molecule here. Now, why is it necessary that there should be these immunoglobulin folds? Is because now where do you find these immunoglobulin molecules? Either as cell surface receptors or in circulation as antibodies. Now, there is a lot of, now even these cells are bathed in the serum, I mean blood, plasma, or in body fluids, all of which contain large number of proteases. To enable these molecules to survive the proteases, nature has allowed the evolution of such molecules which are very stable to proteases. So, you have now again 1/4 of the molecule is the variable region, and the rest of the molecule is more or less constant, and this definitely then should have common functions, biological activity. Let us look at that. But this is one more structure which is based on crystal structures that were obtained for all the monomer proteins, and it was seen. Now, this is very characteristic, and I would like to spend a little time on this. Now, the immunoglobulin fold, each one of this is a fold, and this is the structure of the light chain. You can see constant light and variable light. Now, very clear here are the presence of these anti-parallel beta pleats. These are held together as a barrel, and if one can think in terms of roof of the barrel and the floor, one can see that the roof and the floor are again held together by the presence of that disulfide bond, which I had pointed out in the previous picture. Now, these anti-parallel beta sheets are joined together by these loops, beta bends or loops. And as one can imagine that it is the loops which constitute the antigen binding pocket. Now, this is the variable domain and these are the antigen binding pockets. What is very interesting to see that the immunoglobulin molecule does not have any helices, total absence of helices. There are 5 classes of immunoglobulins or antibodies. Are these similar? Are they identical? Obviously not. So, where are they different from each other? Before we come to the molecular level, let us just look at the domain organization, and IgG and IgD are very similar to each other. The difference lies in the hinge region. Now, the monomer of IgG, IgD and the monomer of IgA are very similar. They have the heavy chain which has variable 1 domain and then constant 3 domains. You can see. However, the remaining 2 classes IgE and IgM both have 4 domains, and what you might see very clearly here, they do not have the hinge region which the other immunoglobulins have. There is lack of the hinge region. In fact, the hinge region in IgE and M has been modified to yet another constant domain. Therefore, now IgE monomer and IgM monomer are longer, the heavy chain is longer than the heavy chains found in the other 3. Is there any similarity between IgA and IgM? Yes, both of them can oligomerize, and the oligomerization, you can see 2 molecules of IgA forming a dimer. Now, IgA can also form a trimer. The dimer and the pentamer of seen in the IgM are held together by the J chain, which is a 15 kilodalton cysteine rich molecule, which is also synthesized by the B cells. You can see the J chain here and the J chain here. So, you have similarities between these 2 molecules because of the presence of J chain. Let us see if there are any other similarities. Yes, now let me come to the subclasses of IgG. I have already discussed this with you earlier, that IgG is present in the serum as 4 distinct subclasses: IgG 1, 2, 3 and 4 in human, IgG 1, 2A, 2B, 3, and these are also present on memory cells as the antigen receptor. Remember that there are constant domain regions in the immunoglobulin, the gene itself, okay? And these not in the sequence, but these are found soon after the gene for IgD. So, you have IgM, IgD and then you have the IgGs, the different domains of the 4 types of IgG, IgG 1, 2, 3 and 4. Now, if you look at these 4, immediately you can see IgG 3 is different because it has a very long hinge region. Hinge region is that region which joins the constant domain 1 with the constant domain 2. Now, there can be as many as 13 disulfide bonds in the extended hinge region of IgG3. IgG2 and 4 are similar in their hinge region disulfide bonds. All of them again would be similar with respect to the domains. They have only 3 constant domains in the heavy chain. Where do these subclasses differ from each other? The difference lies mostly in the amino acid sequence, which is in the hinge region, and a little bit in the Fc region. Now, therefore, one can have reagents or antibodies which can recognize IgG1, but not the others, and so on. So, one can generate isotype specific antibodies. Now, these are isotypes of IgG. Something very interesting about the IgA molecule. I will be coming to that a little later, but IgA molecules are present mostly, the dimeric form is present mostly in the body fluids. Therefore, the molecule has to transcytose across or travel across the epithelial barrier to go on the other side. How is this achieved? Now, the transport of the IgA across epithelial cells was discovered a little later. If I remember correctly, it was sometime in the early 90s. Now, how does this happen is what I will tell you, that you have now the the plasma cell which synthesizes IgA. IgA dimerizes by the J chain. Now, this allows accessibility to a region on the Fc portion of the IgA, one of the monomers, to recognize or bind to a receptor which is also a member of the immunoglobulin superfamily, which is known as poly-Ig receptor, which is present on the basolateral surface of the epithelial cells. So, this is IgA synthesized, the dimer now binds to the polyimmunoglobulin receptor, triggering receptor mediated endocytosis of the entire complex. In the vesicle now, this is a vesicle where there is cleavage of the immunoglobulin receptor. This is the site where cleavage takes place, and now the cut end, the loose end, can wrap itself around the entire molecule, finding a binding site on the other dimer, the other monomer of the dimer. Now, this complex through receptor mediated exocytosis is thrown out, and you have now this molecule of the dimer of IgA, which has the J chain and the secretory component. Now, the same polyimmunoglobulin receptor on wrapping itself around the dimer is known as secretory IgA. This is now, what does the secretory component do? As the receptor, it helps the transcytosis, and after it's secreted, you have the wrapping of this secretory chain, which is around quite large. It's about 70,000 Dalton protein, and this can further add to the stability of the dimer, stabilizing the protein against proteolytic degradation. I'd like you to look more closely at the immunoglobulin that is secreted, the IgA that is secreted. You have the dimer formed by J chain and then the secretory component. Now, this is something quite interesting, though we know that in the serum is the IgG, which constitutes major part of the immunoglobulins, but it is the IgA which is secreted at the rate of something like 5 to 15 grams in humans, but most of it is secreted or transcytosed across epithelial barriers into body fluids. Now, IgA constitutes only 10 to 15% of the serum, and in the serum or blood, the protein is present as a monomer. As an oligomer, either dimer or trimer, IgA is present in milk, saliva, tears, mucus of the gastrointestinal tract as well as urogenital tract as well as digestive tract. Okay. So, therefore, this in fact protects the body surfaces from pathogens. Let us talk now about IgE. When we think about IgE, we always think in terms of the hypersensitivity reaction that IgE mediates. Now, that is a separate lecture, but let us look at IgE which is present in the or secreted into the blood. Average serum concentration of IgE is only 0.3 micrograms per ml, because as soon as IgE, as soon as a memory cell now upon trigger or upon activation by the antigen undergoes class switching, and if it is an allergen and the class switching is to IgE, IgE synthesize is immediately captured by specific high affinity receptors present on mast cells, and this is the picture of the mast cell, basophils as well as eosinophils. And IgE may not have a very long half life in circulation, but present as bound to the Fc receptors, IgE molecules can be stable for weeks. Cross linking of such molecules by the allergen will induce cross linking of the Fc receptors which are present on mast cells, and this leads to degranulation and release of the pre-formed granules, stored granules, components of which are histamine and several mediators, which we will again deal with later. So, this is what IgE does. You might remember that both IgE and IgM have 4 constant domains. Now, this is a graph just to tell you the concentrations, the circulatory levels of human IgE. Now, there has been a little bit of a shift of these the writing here. The first one is IgG, the x axis has all the immunoglobulins. The first one IgG, the green is IgM, this is IgA 1, 2, the monomer, the dimer, this is IgD and this is IgE. Now, the y axis says immunoglobulin levels milligram per ml, but I would like you to look at the numbers. We start with the highest number, which is 10 mg per ml, which goes down to 1, 0.1, 0.01. So, this is increments of tenfold. So, you can see the concentration of IgE, which is this, as being not more than 3 micrograms per ml, whereas IgG in circulation is about 13 milligrams per ml. So, IgG is maximum in the serum. What are the properties of the different classes of immunoglobulin? I will just point out a few. Now, this is a table that talks about the molecular weight and the other properties of the subclasses of immunoglobulin G, IgG 1, 2, 3 and 4, and you can see that all of them are around the same molecular size. I should have in fact said around 1,50,000. The normal serum levels are IgG 1 is maximum 9, 3, 1 and IgG 4 is very low 0.5 milligram per ml. The half life of IgG in circulation is 23 days. This says days, I missed that. Half life in vivo days 23 except for IgG 3. Now, you can immediately link this to the presence of that very long hinge region which allows proteolytic degradation. So, IgG 3 has a stability of only 8 days. Can all of them activate complement? Now, that is going to be my next lecture. IgG 3 of the classes can, but you will see that IgM does that much better, but you can see IgG 1 and 3 do, IgG 3 does that better and IgG 4 does not activate the complement. Do all of them cross placenta to confer immunity to the fetus? IgGs can, but IgG 2 has minimal ability to cross the placenta. IgG1 and 3 can bind to the Fc receptors on macrophages. This allows antibody dependent, the antibody recruiting the cells of the innate immune response. You may have studied that in some of your earlier classes. None of them are present in body secretions. None of them can induce mast cell degranulation, which IgE can. Now, properties of the different classes of immunoglobulins other than IgG, that is IgA1, IgA2, this is a monomer, this would be a dimer, trimer, IgM, E and D. You can see that IgM being a pentamer along with the J chain has a molecular size which is around 900,000. IgE, because of an extra constant domain, has a molecular size larger than IgA1 and IgD, which is 1,90,000. IgA1 is present in 3 milligrams per ml, IgM half of that, 1.5, and IgD, as I have told you several times, that IgD is mostly present as cell surface receptor and almost no antibodies in circulation, which represent the IgD class. Half life in vivo, all of them have much shorter half lives than IgG, which I told you is 23 days. These are only maximum 6 days and Ig is only about 2.5 days in circulation, but remember on the mast cells it is few weeks. Activation of complement, only IgM can activate complement from this group, IgG can, as I told you while referring to the earlier slide. None of them can cross the placenta, only IgG can. Only IgM can bind to the Fc receptors on macrophages or monocytes. IgA and M can be present in body fluids, definitely the dimer form of IgA2, and except IgE, none of them can induce mast cell granulation. Now, we have talked about the structure of the immunoglobulins. What are their functions? Since we have been discussing so much with respect to antigen binding and specificity, obviously that should be the first on the list. And let us begin with the molecule as it is made as a cell surface receptor. Now, immunoglobulins are antigen receptors, they are present on naive and memory cells and they are associated with immunoglobulin alpha, immunoglobulin beta for signaling. Now, this signaling can happen only because the antigen receptor can bind very specifically to the cognate epitope on any antigen. The plasma cells secrete antibodies. The secreted forms of immunoglobulin can form complexes with antigens and these are very nicely cleared by phagocytes. The secreted forms can activate complement cascade, and like I said, my next lecture is going to be that, and the complement activation is a very important part of the immune system. Again, one part of that complement cascade recruits proteins which otherwise would have been proteins of the innate immune system, which is now recruited to the acquired immune system. So, IgM can, then IgG3, IgG1 and 2, these molecules can activate the complement cascade. IgA2 can confer immunity in the body fluid by transcytosing itself across epithelial membranes. IgG1, 3 and 4 can travel transplacentally to confer maternal human response, human immunity to the fetus. The Fc receptors Fc gamma R1a, which are present on phagocytes or macrophages, now can bind to antibodies which have in turn bound to pathogens. Now, once such a complex is bound to the Fc gamma R1a, this activates the macrophages to internalize the pathogen. Therefore, antibodies here by binding to Fc gamma R1a induce phagocytosis by macrophages and granulocytes. Antibodies can also by similar method bind to NK cells, and now NK cells, here in case of the phagocytes, it is phagocytosis which is enhanced. In case of NK cells, toxicity is increased by ADCC, which is antibody dependent cellular cytotoxicity, because these cells have Fc receptors for IgG, and again by a similar mechanism can now bring the pathogen closer to the cells. The linking would be through IgG. I already told you that IgE brings about degranulation of mast cells, basophils and eosinophils, thereby again contributing to the immune system or the humoral immunity. Now, what I talked about with respect to antigen receptors a little while ago is that antigen receptors can recognize, bind and internalize antigen. Now, what I talked about only was recognition, but remember that binding of the antigen triggers the cells to internalize the antigen, where peptides are generated in the cell in the lysosomal compartment, and these peptides can be presented on MHC class II for activating T cells. Thereby, antigen receptors are now allowing B cells to become antigen presenting cells. Can immunoglobulins themselves be antigens? Yes, immunoglobulins are glycoproteins. They are very complex structures, as you have seen so far, and therefore, they themselves can evoke an immune response by way of dictating B cells to make antibodies to them. So, what are the regions on the immunoglobulin molecules which can be antigenic? Let us come to the isotypic determinants. Now, if one purifies let us say IgG from human and injects into mouse, antibodies will be made to the isotypic determinants, which are the differences or the determinants which are present on mostly the constant domain of the immunoglobulin. Now, here the example was human immunoglobulin. So, therefore, those antigenic determinants that are present on the constant domain of the human immunoglobulin. Now, so antibodies would be made by one species to the immunoglobulins of another, and those would be called isotypic determinants. And you all may have used the secondary antibodies, what we call as secondary antibodies as reagents in ELISA, Western blotting, where we use secondary antibodies are nothing but let's say if you are using the primary antibody is mouse, then you would be using rapid antibodies to mouse immunoglobulins, and therefore, those rapid antibodies would be binding to such determinants. What are allotypic determinants then? Allotypic determinants are those that have arisen during evolution. Now, though we say that the constant domain is exactly the same, all IgGs, let us say all IgG1 from the human species would be identical, yet during evolution there is every chance that there could be one amino acid mutation. Now, if those mutations 1 or 2 lie in antigenic determinants, that determinant now would become antigenic, which would be different from the isotypic determinants. Now, there are these allotypic determinants that have been identified, and let me elaborate a little further. When would you see generation of an immune response to allotypic determinants? In blood transfusions. It is very apparent that normally we should not be making antibodies even if we receive antibodies from other members of our species. But in blood donation, people found that there was a reaction or antibodies were made by the recipient or the receiver from the donor's immunoglobulins, and on closer look these allotypic determinants were identified. What are idiotypic determinants? Idiotypic determinants are those which are present on the variable region. Now, as is shown here, the idiotopes. Now, idiotypic determinants would be those that would bind to that region or an immune response that is generated to that variable region, and in fact, which I haven't mentioned earlier, that these idiotypic antibodies which can be generated can also regulate the immune response, not can, do regulate the immune response. Idiotypic antibodies can be shown very easily in an experiment. In fact, you can think of doing that experiment. A rabbit which is injected with one antigen, you will see no idiotypic antibodies made in the first two, let us say, exposure to the same antibody. Now, let us say the rabbit has received ovalbumin, there will be antibodies to ovalbumin. In the first generation, the primary dose, the secondary immune response also will give you mostly antibodies to ovalbumin. But let us say from the third immunization onwards, you may start getting antibodies to the antibodies to ovalbumin. And these in fact are regulators of the immune response, not allowing the immune response to go berserk, and there is a decrease in the amplitude of the immune response. So, antibodies to idiotopes can also have several uses apart from the regular immune response, which dictates the suppression to some extent of response to the primary antigen. We will come to that a little later. So, just to go over the antigenic determinants on immunoglobulins, you have isotypic determinants which define each heavy chain class and subclass and each L chain type within a species. Within a species, each normal individual will express all isotypes. This is important. However, allotypic determinants, some alleles encode subtle amino acid differences in a few members of a species. Now, these are the ones that have been are well known, and therefore, I have written them here. In case of human, the gamma chain allotype, there are about 25 such allotypic determinants, and also on IgA there are 2, and kappa light chain there are 3. Idiotypic determinants, I have already said, are those determinants that are formed because of the presence of unique amino acid sequences of the VH and VL. We should also think about immunoglobulin molecule as being a glycosylated protein or a glycoprotein. So, there has to be some role that glycans play. It was thought that since immunoglobulin, when it is secreted in B cells, it is secreted as heavy chain and light chain, and these have to come together, and then when they are held together, disulfide bonds have to be formed, and now these are exported outside either as cell surface receptors or secreted as antibodies. Therefore, the glycosylation is doing, its function is only to help the protein folding, but we know that glycoproteins, the glycans, also stabilize the molecule from or protect it from proteolytic attack. The involvement, the importance of these sugars has become now known as people have, glycobiologists have started looking at the glycans, and I would like to just mention here that there are about 30 different glycoforms of immunoglobulins, and some of these glycoforms are conserved, others are highly variable. This glycan chains which I told you are present on the FC region, the CH2 region of the FC region. These molecules, the glycans, are very important for the immunoglobulin to bind to the FC. I have just told you about the importance of these FC receptors, and it is the sugars which allow this linking. Decrease in the glycan structure now does not allow a tight binding of the immunoglobulin with the FC receptors. Also, the sequences of these oligosaccharides, the sugars that are present, has been shown to inhibit an autoimmune response, the autoimmune response which might result in rheumatoid arthritis. Now, let us see how that happens. There are people who have the condition of G0 glycoform, that is, they do not have those enzymes which transfer galactose, terminal galactose. Therefore, if in the absence of galactose, there is exposure of N-acetyl glucosamine. Now, this allows binding to the MBL which is mannose binding lectin, and we will be discussing this what MBL is in the next class. And this MBL, it is a cascade of the complement pathway which can lead to inflammation and rheumatoid arthritis, one type of rheumatoid arthritis. Now, what I would like to do is stop here and then begin my next lecture with two immunoglobulins that I have not discussed with, but they are quite important, especially one of them the single chain and domain antibody, which is known as Campbell antibodies, because this has helped molecular biologists to be able to express only the variable domain of the immunoglobulin, and these are known as single chain variable fragment, and nature has done this in camels, where the single chain, I mean the antibody, 30% of the antibodies in camels are made up of only heavy chain. So, I will stop here, and in my next class I will start with the introduction on the camelid antibodies as well as the IgY antibodies. Now, why IgY antibodies, which are from the avian system, why this is important is because these, they can be used much in diagnostics. Also, immunoglobulins made by birds is deposited in the egg yolk, and therefore, it is much easier to be able to purify antibodies from the egg yolk rather than from the serum, because the presence of large amount of albumin. More importantly, eggs have something like 3 grams of immunoglobulin Y, no sorry, not 3 grams per egg, but 3 grams of immunoglobulin Y can be got from eggs in a single month from one hen. So, I will talk about this as well as I will introduce you all to the complement cascade. Thank you.